agenda. We originally had Dr. Paulo Shea, but it did come to our attention. Unfortunately, there's an emergency that he is dealing with. So, in his place, from his sister company, from Crystal Pharmatech, we have Mr. Robert Winslow presenting from Crystal Pharmatech next. And yeah, the floor is yours, Robert.
Okay. So yeah, I'm a fill-in. I just found out about, I guess, less than an hour ago that Paul couldn't make it. So this, I don't know how it fits in with your agenda. I know I scanned through, I saw several people that I've worked with in the past, including Margaret Offerman. So I think this is probably a decent venue. If not, I hope you enjoy the talk. This is something completely different, I think, towards what the agenda was on for today. Yeah, so my background is I spent 15 years at Merck Research Labs, involved with salt selection, form selection. And what we started at Crystal Pharmatech about 10 years ago is a CRO that was dedicated to selecting the right form and formulation during every stage of development. And this kind of starts with the first time you put your fingers on the molecule all the way up until past approval. So people like to think about salt selection and formulation as a unit operation in their CMC document, but it's really an ongoing process. And it really starts from literally the first time you touch the molecule. And so we've kind of come up with a mission statement here based on our experience within the company and our network within the solid state research realm. And some of the key takeaways with the mission statement is that we really try to make best-in-class decisions with a multidisciplinary approach. And what that means is getting input from not only medicinal chemists, process chemists, engineers, but formulation as well as DMPK. So it takes a village to raise a molecule, and understanding the impact of not only form, but also formulation on different different departments and different disciplinaries really is impactful. And we really rely on fundamental solid state data, and I'll go into some of that. You see pieces of information like solubility and stability, and you'd think solubility is pretty easy to measure, but it's actually a pretty complex analysis when you get down to understanding the solid form and potential disproportionation, salt formation, co-crystals, etc. And what we're trying to do here is right first time, fast first time. And we work with a lot of smaller person in a molecule companies or smaller biotechs, mostly with small molecules, some peptides and small proteins, basically getting you to the goal line the fastest, best way possible. And so this is a little background of what's the impact of form and formulation. And you can see this is even before candidate selection. And choosing the wrong form or formulation can really lead to a large delay, program delays, and obviously comes down to money, money, money. And I know a lot of companies early on will try to dose animals and get the molecule in the solution any way possible. And we're not saying to do your salt selection way back in the lead optimization space when you have maybe 10 lead compounds. That's not what we're saying. What we're saying is you at least want to know what you're dosing. What's the form that you're dosing? What are the vehicles you're using? What is the visibility into long-term tox studies? And what's the visibility and the liability of that form? And so some very simple information on maybe 10 or 15 milligrams of material, you can tell if your material is crystalline, if it's potentially a salt, what its melting point is, maybe even get an accurate water solubility measurement. And all of these small pieces of data create a picture of your compound as it goes through development. So just dumping a white powder in DMSO and shoving it down a rat's throat is not a good way of getting early PK studies. You really want to understand your form and formulation at every stage of development. So what is Crystal Pharmatech? We've been around since 2010. We have well over 700 clients worldwide. We're a global organization with research facilities in China and in Cranberry, New Jersey. And we've supported well over 1,500 drug molecules. And back at Merck, we were working on about 40 preclinical candidates a year. So we've seen a lot of different types of compounds with a lot of different types of issues, whether it be solubility or melting point or whatever physical malady your compound may have. And so we also, mentioning Toronto, we have another sister organization in Toronto, a formulation site called Can-Do Pharmaceuticals. And so what we're trying to have here is kind of a highly technical one-stop shop to get you from candidate selection all the way to first in humans, your entire CMC package, making you GMP API, making you your GMP formulation, getting the right formulation, not just throwing the API in a capsule, looking at the formulation towards the long-term formulatability of the compound. And what we offer over the larger one-stop shops is we each have our specific expertise in our certain areas, in the API area, in the formulation area, and the solid form area. And we really cater to the smaller biotech type companies and can provide that guidance, not just data, but guidance on how to develop your compound. So Can-Do in particular is our formulation arm, and they have a lot of activities in formulation development, and bioavailability issues. They have spray dry dispersion capabilities, hot mill extrusion, but basically a lot of times you don't necessarily even need those. And so it's really understanding the target product profile of your compound and knowing the true BCS classification to understand what type of form and formulation you need for development. A lot of times clients will come to us and they say, well, I need a co-crystal or I need a dispersion. And after a decent amount of discussion, you realize that the right PK studies really weren't performed, and they had a consultant that told them that the very low solubility compound and it's going to be a BCS4, when in actuality they didn't have that data, and they kind of backed themselves into a corner in trying to get a dispersion when in actuality they could get away with a standard solid oral dosage with the right excipients and the right formulation. And so we often turn away a lot of work just for the fact that a lot of times the work isn't necessary and the problem is somewhere else instead of with the crystal form. And that's kind of what we offer in combination with Can-Do and our API manufacturer here in the States. And so if you're looking at properties of the API and formulation throughout the development cycle, and lead optimization, where you have maybe 3 to 5 compounds, you're not sure which one's going to be the best for development. Obviously, you're worried about safety and safety and does it work, the efficacy of those lead compounds, and those are the top kills. I'm not saying that they're not, but with a little bit of information, you can get an understanding of developability. And the developability, if everything else is pretty much the same, can really help you get a compound that's going to be a straightforward development. And this type of fundamental data early on, you're talking about a good pKa to know if you could form a salt or not, good log P to determine permeability, pH-dependent solubility. And these aren't just check boxes. We're going to take this information and we're going to actually work with the compound to formulate it in a way that's going to get you your consistent exposures in your animal studies. And so we really want to take this away from a checkbox type of research and development and really put it into a full understanding of your compound. And so I'm not going to go through all of these bullets, but basically this is a stepwise process getting input from the appropriate experienced disciplines that'll get you the right API phase and formulation all the way up to first in humans and beyond. So, what specifically are we talking about core competency wise? Very early on, it's the pH solubility, tox and PK vehicle development, looking at the appropriate salt form. Should you go with the free, crystalline free form, an amorphous form, or do you need to go for a salt to increase your solubility and increase your exposures potentially? Do you have to mill your compound? Can you control the particle size in your crystallization? And then past the candidate selection stage, you want to start thinking about your scalability of the form and developing that last step isolation and understanding the potential impurity crystal forms that can pop up in your crystallization. I know early on, a lot of the API is isolated through column chromatography and then either lyophilization or just rapidly pulling off the solvent, which typically can lead to amorphous material. And that's okay. I mean, as long as you understand that amorphous material, the hygroscopicity, what it likes to crystallize into, because when you put that amorphous material in a vehicle, like a methicel suspension for dosing, 90% of the time that's going to convert over to something with a lower solubility, and your exposures are of that crystalline form and not of the amorphous form. So just small things like that about understanding your form and formulation can really go a long way towards having a robust process. From a formulation aspect, we have Can-Do has some proprietary technologies that can really bump up your exposures, and I really can't get into it due to the proprietary nature, but basically formulating around liabilities of the API. And so we're not, so there's a lot of competition out there, obviously, and solid state characterization, crystal form, pretty much everyone says they do salt selection and form selection. And we're kind of different. And what makes us different, we feel, is the way that we attack the understanding of the form. And we really have old school, what's called point of observation driven culture. So a lot of times you'll get these 400-page reports about a salt selection, and it looks like you have 30 forms, and you've got all types of X-ray, all types of DSC, and you really don't know what to do with all that data, right? And so what we try to do is we try to figure out the variability right at the spot of measurement. And so, for instance, if we're taking an X-ray and that X-ray doesn't match immediately, we're gonna at that point understand what's happening. We're gonna determine to see if that is what's called preferred orientation or an impact on the X-ray from the particle size or particle shape. We're going to try to grow single crystals to get good reference spectra. And so we're trying to understand right at the point of contact what's happening with your compound, how many real phases do you have? And we don't assign crystal forms until we really understand what they are. So you'll get reports back and you say, well, I've got 17 polymorphs, when in actuality, you might only have 2 types of families of solvates and hydrates, et cetera. So it's that full understanding of the entire solid state package around the crystal forms that really separates us out. And, we're focused on what's going to get your process to the next step, right? So, we're strong in the science and we, co-crystals, twinning, whatever, but, we want to get you from point A to point B. We want to know what the limitations of your API are, what has to be involved with your formulation development, and what are your risks with your phase going forward. So just a couple examples of our capabilities. This was a case where, as I mentioned, the CRO before us had said that the polymorph system for a free base was much too complex, and so they wanted to switch to a salt. The only reason they wanted to switch to a salt was because of the complex polymorphism. They didn't need it for exposure. They didn't need it for their formulation. And so to try to avoid that, because a lot of times, like I'm mentioning, people will think they need a salt or think they need a dispersion, when in actuality they haven't done the appropriate research to understand what they really need. And in this case, it wasn't that complex of a polymorph system. It was just that they were labeling polymorphs what weren't polymorphs, and it wound up that it was actually a bunch of solvates, and all we needed to do was find a solvent where you weren't solvating, and then if you isolated or crystallized your material from that non-solvating solvent, you could not direct the polymorph, but you'd get out your most desired polymorph. And so, you can see rather quickly how we develop the crystallization, save the client a ton of money from trying to go to a salt selection and maybe even delaying their GMP delivery, because the API manufacturer was already developing the GMP process to isolate the freebase. And so we stopped that delay. We were able to get a solvent system where they could recrystallize and get out the crystal form that they wanted. They weaned off the other CRO, and we successfully got to scale up the GMP campaign in August of the appropriate crystal form. And again, we treat form and formulation as in a staged process, and so not doing upfront work, so to speak, right? So early on, and we break this up into stages, and you're just trying to get from point A to point B, the minimal amount of data to know your risks and to get you from point A to point B. And we have workflows basically established for every stage of development from lead optimization. So if you came to see us, in the middle of stage D1 and D2, we've got the appropriate workflows and the appropriate vehicles to test and the information that's needed to get you to that next step. Not just us, but we also have a scientific advisory board, and also have a panel of consultants that we can specifically bring on to very difficult programs. And these consultants, again, are varied disciplines, from API development to formulation to engineering and salt selection. We also do spray dry dispersion. Again, we're a small scale non-GMP Crystal Pharmatech, and so we can evaluate dispersions in a very quick time frame. And a case study here was for a client that wanted to evaluate a bunch of, basically a lot of their compounds, evaluating dispersions in DMPK studies. And so from delivery of API, they wanted in a 2-week time frame to be able to choose a spray dry dispersion, actually make the spray dry dispersion for the DMPK study. And so we can, we have the flexibility and the speed and the solid state understanding to be able to quickly develop spray dry dispersion formulations when they're needed. So really, why us? And summary is that we use a multidisciplinary approach. We come from, different areas of understanding to get input from all areas on how to develop the compound. We have this point of observation, understand why culture, fast and flexible. And the key is that we're driven by sound solid state fundamentals. So at that, I'll take questions and thank you very much for your time.
Thank you very much, Robert. And once again, thank you for on a short notice doing this presentation. I know Dr. Paul is going through certain issues and our hearts are with him, definitely. And definitely, if anyone's interested in connecting with Robert and his colleague, John Balesta, feel free to send us a message if you want to learn more about their facility in Toronto, which is right near our office as well, coincidentally. Um, feel free to let us know. So once again, thank you, Robert, for your presentation.